Hey guys, I'm back with another journal article. This one is called Ubicome, Hybrid Deep Learning Model for Predicting Plant-Specific Protein Ubiquitination Sites. And this journal talks about the utility of machine learning models, specifically AI networks, and combinations thereof, which we'll talk about by the end, and how this has led to the most recent advances in AI technology and its utility in a huge range of applications, and in this case, it's a really important one about ubiquitins. So, a word about ubiquitins. They're a thing in biology that are present in all living things. Um, it's basically a little protein that connects other proteins. It's almost like it's kind of like a vehicle of connections between proteins, like proteins where it is, where it's connected to other proteins, and like how it facilitates cellular function is it's all bound up in these ubiquitins. So they have three enzymes. They have one for attachment, one that they actually transfer, and then or connect, and then one for the transfer of the residues, they call them. So it's an amino acid, and they're utilized by your body's inflammatory response system. They're utilized in therapeutics. They're also cited as source problems um, for when they don't work right. They've been linked to a lot of different diseases, neurodegenerative issues, cancers, immune problems, etc. But the most interesting and important thing I would guess about our knowledge about ubiquitins is they're um, highly dynamic and reversible because they're responsible for all of this mediation of protein attachment. So the what I'm about to talk to you about is from this journal that you can find online. I'll leave a link in the show notes. And it was a really transformative article because it utilizes a combination of neural networks, two different types of neural networks that really helped us get into stuff that everyone's using now, which is more like transformer models. So the challenges that they were attempting to solve using neural networks was that the thing about ubiquitination, which is the process of identifying these sites, was that it's very expensive and very time consuming. It's a very tedious task in trying to identify these sites of these proteins, or these residues, and on the proteins, or connecting to the proteins between these cells, like in plants and in food, human bodies, and everything in between. So it's a super expensive and laborious process, and it's been an ongoing challenge to try to determine those sites effectively. So up until now, we've been using handcrafted features, which I will mention in a little bit. But first, I wanted to show you a little bit about what that ubiquitin looks like. So this is a graphic of a ubiquitin. And so basically, it's these little gold squiggly lines that you see are the lysines, and that's how they attach to proteins. And so they're kind of like these intermediaries between proteins. So this is the model architecture that the authors proposed. And as you can see, it has two modules. And I'll go into detail about each module. And then at the end, they combine to make one final module, and they have only two outputs. So all this complexity for just a yes or no, this is the path of this amino acid. So we're going to talk first about the first module. So what they did is using a long short-term memory networking layer. First they started with sequencing the ubiquitination sites. As you see on the left of the graphic, This is from their article. So first they identify all of the sites that the ubiquitination occurs. So this was actually done using real plants for this article. And then they introduce in input one. You see they introduce index-based encoding to represent the protein fragment residues. So basically, that means they're assigning an integer to each of the sequence identifications, each of the sites that they've identified. So, and then the most important part of this is the embedding layer. So that's why they're calling this embedding encoding module. So the embedding is a vector that they assign a 32-dimensional word vector, and that's assigned to each site and its relationship to that particular surrounding connections. So as you saw from the graphic, as you saw in the graphic, where there's different lysines that attach to different locations. So all of those need to be kind of mapped. So the sequencing is a way to keep everything in order, basically. So the semantic correlations are being captured by this embedding encoding schematic that they've figured out by basically keeping everything in a layered sequences, so that they could track which connections are all together and most important. So we'll get into that a little bit more with the next module. But what was really cool about this first module is that they were able to eliminate the handcraft nature of all methods that had been used so far with just simply statistical and machine learning, and now we're able to use backpropagation and automation methods for the algorithm to actually detect for itself, using weights and biases and backpropagation, of where the most important connections are and where the end connections are, like end to end. So basically where one amino acid stops and where the site of another one starts. So that was really monumental for this medical procedure, for it to be automated in this way. So the next section is we're going to talk about module 2. So this other module, which was kind of happening in parallel with the first, they call it the physicochemical properties module, and basically there were 237 properties, and they gave a semantic description for 20 natural amino acid residues. So using those descriptions, they were able to assign 5-dimensional vectors for each amino acid residue in order to describe its properties. So it's the properties and their connections with each other using cosine similarity and the sequencing, most importantly the sequencing, that they were able to determine connections and identifications. So this is really monumental for the identification part. So as you know, like the kernel filter in like a linear binary, linear binary programming, you could take like a 3x3 matrix and you line it out to kind of like this one on the right side, a one-dimensional vector, and when you put that into the ones and zeros, then you compare it to the middle pixel, the 9th one, and then with those 8 ones and zeros, that's a binary 8-bit binary number integer that you get because each placeholder is an exponent for 2. So, or as an exponent, exponent. A placeholder is an exponent for 2. That's right. And so, using those those sequences and kind of overlapping them as they're used like as a sliding window, they're able to retain the connectability of um, of these properties because and then kind of keep it going as a tag like for each for the 5-dimensional vector per amino acid. So um, they used a convolution network for this. That's the second grid, and the third grid is going to be the max pooling, and then finally it's flattened out and delivered to the end product. So the max pooling is basically the scaling down of features, so that using similarity measures, they're able to eliminate and discard stuff that's not going to be as important for the weighting mechanism in backpropagation. So um, at the very end, we get to the concatenation process, and this is the final output, the really cool thing. So in this spot, both of the modules are concatenated, and they each of them containing the feature vectors. They're passed to the final deep layer for the deep feature extraction, and then using typical regularization methods. This is just a neural network model basically, but it had used the long short-term memory and the convolutional neural networks for the second one, and concatenated both of those into one final artificial neural network. And this is and then it's having applied regularization methods to decrease bias and variance as much as possible. And the hyperparameter tuning was so that they could find the best optimization methods for the model to keep it from overfitting and decrease the bias as well. And then the softmax output is to determine basically whether it's a plus or a minus or connection or not. So like is this continuing on the same site, or are we now onto the site of a new amino acid residue? So it's the identification mechanism. So the implications that the results of the testing were improved about 80% or more, which was about an 8% increase from previous models, but most importantly, it was just much more efficient, much less costly, and more less time consuming. So the implications of where this is going and where it's going to continue, already we know with transformers, there's even more technology out there when it comes to neural networks that's already getting implemented at a wide scale. So this really was kind of the dawn of the age of the transformers. So, but really cool, monumental to kind of be that stepping stone between the CNN models, the LSTM models, and now what we have is transformers. So, which is really just kind of the next step. So the implications already when this paper came out were so important because it really increased the site detection of these protein locations. So it already was able to increase the efficiency, reduce cost, and bring about more advances in medicine more quickly. And then, of course, because of the lower cost and the efficiency, it was able to save money for everybody. So it's just a really cool paper. I invite you to please go take a look for yourself and read it. It's a really easy read, and it's a really cool. They even list out the kind of neural network that they used, the layers, the convolution layers, and you can find some code as well that they have there publicly available to use it for your own applications. So thanks you guys for listening, and I'll see you next time.